correct chapter here, and we're on the chromosomal mutations. So when a segment of DNA is copied multiple times, this is duplication. Yes. You have A, B, B, C, and then D, F. So the B is duplicated. What is when the segment of DNA is reversed? This is inversion. Yes. What is a chromosomal inversion, in which a segment of DNA is swapped with a segment of DNA from another chromosome? This is translocation. Yes. The Griffith experiment showed that bacteria can get DNA through a process called transformation. R is safe, S strain is deadly. And that happened with mice. It did have a good image of it, but I'll show you. I'll put it under this slide too. I think this is what also gives them the antibacterial resistance. What experiment built upon Griffith's experiment showed that DNA is what caused the bacterial transformation? I think this should be the next one, which is the Avery-McLeod-McCarty experiment. This brought us one step closer to proving that DNA holds our genetic material. So they discovered that it is the DNA. It's called DNA. It is the characteristics of DNA. Hypothesis: the genetic material of the cell is either protein or nucleic acid. So DNA or RNA. Remove lipids and sugars from a solution of heat-killed S cells. Proteins, RNA, and DNA remain. Treat solutions with enzymes to destroy protein, RNA, or DNA. Add to culture containing living R cells. Observe for transformation by testing for the presence of virulent S cells and heat-killed S cells. Lipid, sugars. Add proteinase, no protein. S cells appear. Add RNase, no RNA. S cells appear. Add DNase, no DNA. No S cells appear. So the hypothesis was that the genetic material of the cell is either protein or nucleic acid, DNA or RNA. The conclusion was that transformation requires DNA, therefore it is the genetic material of the cell. How did they have DNAs, RNAs, proteinase back in the days? God knows. We can research. If anyone knows that, you can drop it in the comments if you research that. So remove lipids and sugars from a solution of heat-killed S cells. Treat solutions with enzymes. Where did they get these enzymes from? How did they get it? To destroy the protein, RNA, or DNA. Add to culture containing living R cells. Observe for transformation by testing for the presence of virulent S cells. Cool. And what experiment confirmed DNA is what contains genetic material? This should be the third one. The Hershey-Chase experiment. So it confirmed it, basically. Experiment 1, testing proteins. Protein codes radiolabeled. Bacteria infected, no radioactivity enters cells. Phage grown with radioactive sulfur. Centrifuge, radioactivity in supernatant. Is the supernatant the wild type? Phage DNA radiolabeled, bacteria infected, radioactivity enters cells. Phage grown with radioactive phosphorus P32, centrifuge, and radioactivity in pellets. DNA is the genetic material. Proteins are not genetic material. Nice. What is a square diagram that is used to predict the genotype of a particular cross or breeding experiment? This is the Punnett square. Yo, past the p-planes. You better not use a Punnett square to calculate the recombination frequency of the alleles. Who came up with this? A dihybrid cross shown above will result in the following phenotypic ratios. So, expressed AB. So we have AB, 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 and AB like that. Same here. So I think for these, usually it is 1, 1, then you have 14 left, which is down to 7 for this one. Is it 7 for the other one? Wait, so this is A capital, B capital, right? Then this is A capital, B small, or B capital still. This is A capital, B capital. So it's actually 4 and 3, 7 for A and B. So for this column and this row, it's all going to be A, B capital, right? Because they're dominant. Then here it's going to be A big and B small. Sorry, B capital here as well. So that's +1, 8. And we also have this one. So this 9 out of 16 here. We have 5 left, 7 left. So yeah, 9, 3, 3, 1. So 9 for that, and so 9, these two. And for this one is A big and B small. A big and B small. And this one A big and B small. It's only the last one that is AB recessive. And for these, the B is big. So, yeah. When two genes are close together on the same chromosome, they do what and are said to be linked? They cross over. They do not assort independently when they are close together. Oh, when they're far, they do the linking. They're said to be linked genes. So the other ones that are not linked, they do the crossover. They do not assort independently. They assort those are not linked will assort independently. Unlinked genes dihybrid pattern, 4 potential gametes combination. Linked genes monohybrid pattern, it's monohybrid for these. So 2 potential gamete combinations. You get the 9, 3, 3, 1 for the dihybrid. For certain phenotypes may not occur unless there is recombination. Linked genes, for example, on the same chromosome, don't confuse with sex-linked. Without crossing over, how many types of gametes can be made? Just 2. Without crossing over, it's only 2. AB capital and AB small. It's homozygous. Tetrad, crossing over, AB capital and AB small. With crossing over, how many types of gametes can be made? 4. You get AB, so you get the heterozygous as well. Nice. What is the likelihood of two alleles being separated during crossing over in meiosis? Is this the Hardy-Weinberg probability? Recombination frequency? That's the likelihood for two alleles being separated during crossing over. Is likely a reflection of the physical distance between two genes. The further away the genes are, the higher the frequency, right? The higher the recombination frequency, because they're not linked. Genes close together on the same chromosome, gametes made, 50-50 almost between the homozygous dominant and homozygous recessive. But with the recombinant, you get the heterozygous as well. Prophase 1, meiosis 1, crossing over. We know it happens in the prophase stage. The farther apart two alleles are on the same chromosome, the more likely they are to assort independently. Recombination frequency is the reflection of physical. Eye color, body color, wing size. For wing size and body color, it's a higher chance. Nice. Give the Hardy-Weinberg equation. So it's p squared plus 2pq plus q squared equals 1. Yes. This is for the heterozygous chance. And this is for the dominant zygous, and the recessive one is q. The mechanism for evolution is, we had this. It's natural selection. Each species shows variation. Get off my land. There is no competition within each species for food, living space, water, mates, etc. The better adapted members of these species are more likely to survive. Survival of the fittest. These survivors will pass on their better genes to their offspring, who will also show this beneficial variation. So the mechanism for evolution is natural selection. Several major ideas about evolution came together in the early 20th century to form the modern synthesis model of evolution, including 3 of them. This is about the conversion, genetic variation, natural selection, and Mendelian inheritance. So we have variation, breeds, races, subspecies. We have malfusion, competition, geometric population growth, limited resources. Combined together, 19th century, you get natural selection, survival of the fittest. Then we have mutations, small changes in individual characteristics. Then you get genetic variation or Mendelian inheritance. For Mendelian, you have 2 copies of each gene, 1 from each parent. The genetic variation, alleles of individual genes combining to give continuous variation. Then you get the modern synthesis, early 20th century hypothesis. So natural selection, Mendelian inheritance, and variation, genetic variation. What theory considers evolution to be a very slow process with intermittent rapid bursts of evolutionary activity? Is this the molecular clock? No, the punctuated equilibrium. Rapid bursts, punctuality, gradualism over time, punctuated. It's just suddenly you get all these things. This is like a conversion thing with gradualism that happens. So gradualism is the opposite of punctuated equilibrium. So punctuated equilibrium. Nice. Okay. Again, yes. Word selection is a type of natural selection in which the population mean stabilizes on a particular non-extreme trait value. This is stabilized natural selection, stabilizing selection. Then we have disruptive and directional. So stabilizing is before here with the red, the after is here with the blue. So the traits will be non-extreme in the middle. With disruptive, it will be on both ends. In the middle, you don't have that much, and directional is one end has it goes to the other side. How does the trait change? Directional selection against one extreme for one extreme trait against the other extreme. For example, long wiggly tails look like a snake and scare predators. The longer the tail, the more it looks like a snake. Stabilizing selection against both extremes. Modern traits against both extremes. Short tails mess up the cat's balance. Long tails drag on the ground. Medium tails are the best. And then disruptive is both extremes. Moderate traits. Short tails help keep predators from catching you on the ground. Long tails are good for balance in trees. So medium tails don't help. So disruptive is the opposite of stabilizing. Stabilizing. Robins typically lay 4 eggs. An example of stabilizing selection. Larger clutches may result in malnourished chicks, while smaller clutches may result in no variable offspring. Then we have this is for directional. Light colored peppered moths are better camouflaged against a pristine environment. Likewise, dark colored peppered moths are better camouflaged against a sooty environment. Thus, as the Industrial Revolution progressed in 19th century England, the color of the moth population shifted from light to dark, an example of directional selection. And third one, this is when you get two extremes, both extremes, you will have disruptive, diversifying shown here. In a hypothetical population, gray and Himalayan, gray and white, rabbits are better able to blend with a rocky environment than white rabbits, resulting in diversification. So we can also name it diversifying. Okay, diversifying. Cool. What selection is a mode of natural selection in which one extreme phenotype is favored over other phenotypes, causing the allele frequency to shift over time in the direction of that phenotype? This is directional selection. What selection will cause organisms with intermediate traits to reproduce less often, and will allow those organisms with extreme traits, this is multiple traits, is disruptive, to reproduce more often. This drives the population apart. Disruptive or diversifying selection. What is the relatively fast evolution of many species from a single common ancestor? Inbreeding? No. Adaptive radiation. Adaptive radiation generally occurs when an organism enters a new area and different traits affect its survival. Think of mammals after all the dinosaurs died, like there was so much space and land. So we have the founder species, and then the adaptive radiation. A single species rapidly adapts to fill available niches in an environment. Seed eaters, insect eaters, cactus seeds and plants, buds and fruits. So what radiation? Adaptive. not active, adaptive. The molecular clock model states that what is related to the what since the two species broke off from a common ancestor. The degree of difference in the genome between the two species is related to the amount of time since the two species broke off from a common ancestor. So the more our genes are different, so with Neanderthals, we don't have that much difference. So the degree of difference in the genome between two species. And then we have Neanderthal ancestors. And then we have chimpanzees that are much more different with us. 9.3, 6.5 million. Neanderthals 50,000 years ago, 40,000 years ago. The molecular clock model. What evolution is the process whereby groups from the same common ancestor evolve and accumulate differences, resulting in the formation of new species. Divergent evolution. So accumulate differences and form new species. This is divergence. Convergent should be the opposite. Streamlined shape due to aquatic environment, shared selection pressure between shark and dolphin, but dolphin and puma are closer since they're bony, mammalian, bony skeleton, lungs and mammary glands, but sharks, cartilaginous skeleton, gills, scale, denticles, and skin, vertebrata, cranium, tripartite brain. Divergent evolution here, but these two are convergent, closer together. We have evolution type. So divergent is ear tufts for trait and species F. tufts and F. gricio, and then convergent is coat color, and then parallel is tail shape. Nice. Then we have what evolution is the independent evolution of similar traits starting from a similar ancestral condition. Is this convergent? Parallel? Similar traits. So independent evolution of similar traits is independent evolution starting from a similar ancestral condition. So tail shape is parallel. This one is completely away from this one, the orange code from the orange code here. But the tail shape is what is parallel between them, right? So independent evolution of similar traits starting from a similar ancestral condition. Mendel's what law states that the alleles of two or more different genes get sorted into independent assortment, right? Sorted into gametes independently of one another. So the pairs of alleles are separated, that's segregation, but independent assortment is that they are independently sorted and not crossed over. The pairs of alleles will be sorted independently of one another when gametes are formed, and then the principle of dominance is at the end when one allele masks the effect of another allele. These laws are known to be due to key events that occur during meiotic division. The law of segregation should happen in prophase 1, right? Describes how homologous chromosomes and hence allele pairs are separated in meiosis 1. The law of independent assortment describes how homologous proteins align randomly as bivalents during metaphase 1. So that's prophase 1 for segregation. And the homologous pairs align randomly as bivalents. So randomly independent assortment, and that's during metaphase 1, where they meet in the middle. Through the elucidation of the process of meiosis, we know that there are certain exceptions to Mendel's laws. Genes that are not the same, that are on the same chromosome, linked genes, will not undergo independent assortment unless recombination occurs. So actually, independent assortment is the same as recombination, right? That's why linked genes will not go under independent assortment. Not all genes display a dominance hierarchy. Certain traits may be display codominance or incomplete dominance. So law of segregation. Each hereditary characteristic is controlled by two alleles, which separate into different gametes. In law of independent assortment, the separation of alleles for one gene is independent to allele separation for another gene, unless they're linked, right? Cool. What is a chromosomal mutation in which a large segment is lost? Deletion. What is the process whereby organisms, while evolution that are not closely related with, will independently evolve similar traits as a result of having to adapt to similar environments or ecological niches? Whereby organisms that are not closely related will, so it's not closely related, it's not divergent, it's either convergent or parallel. So will independently evolve similar traits. When it's independently, it's probably convergent. As a result, having to adapt to similar environments. This is convergent, right? The shark and the dolphin. The coat color is convergent. What is parallel? The tail shape. What is divergent? The ear tufts. You see they have ear tufts and ear tufts. Tail fluff, orange coat. The tail shape is parallel. I think we need to be able to draw this to understand these divergent, convergent, parallel ones. But yeah. Gene environment interaction is when what responds to environmental variation in different ways. Two different genotypes. So gene environment interaction, when your genes are interacting with the environment, is when two different genotypes respond to environmental variation in different ways. Jennifer and Karen, identical twins born with genes that absorb fats twice as fast as the average person. Jennifer eats healthy, low-fat food. Karen eats fatty, unhealthy food. Jennifer obesity genes not triggered. Obesity genes in Karen is triggered. So two different genotypes. Gene environment interaction, responding to environmental variation in different ways. But is it two different genotypes or is it the same genotype? A study that looks at the interaction between genetic predisposition and environmental events is looking at what? Not epigenetics. It's gene It's looking at gene-environment interaction, not epigenetics. Epigenetics would be methylating something. Epigenetics is the mechanism for gene-environment reaction. So the mechanism, like natural selection that we had, the mechanism for environment reaction is epigenetics. It's like methylating the DNA. Give me a second. So the probability for that is 9331. Dihybridization. What is the likelihood of two alleles being separated during crossing over? What is the likelihood of two alleles being separated during crossover in meiosis? It's the recombination frequency. Yes. When two genes are close together on the same chromosome, they do what and are said to be linked? They do not cross over. They do not assort independently. Several major ideas about evolution came together in the early 20th century. These to form the modern synthesis model, that was natural selection, Mendelian independent assortment, inheritance, and genetic variation. What theory considers evolution to be a very slow process with intermittent rapid bursts of evolutionary activity? This theory would be, so with rapid bursts, you have this is about gradualism, and the opposite, which is what equilibrium? This would be genetic equilibrium, punctuated equilibrium. Let's do it again. What is the relatively fast evolution of many species from a single common ancestor? Fast evolution of many species from a single common ancestor, that would be this is adaptive radiation. Nice. What molecular model, the molecular clock model, states that what is related to what? It's since the two species broke off from a common ancestor. So it states that the variability in the genes is related to the degree of difference between genome between two species is related to the amount of time. Okay. Degree of difference in genome is dependent on time. Related to time. So what evolution is the process whereby groups from the same common ancestor evolve and accumulate differences, resulting in the formation of new species. So you have new species divergent evolution, and you accumulate differences. What evolution is the independent evolution of similar traits, starting from a similar ancestral condition? Independent evolution of similar traits, there we have when you get similar traits and independently starting from a similar ancestral condition. Is this parallel or convergent? I think it's convergent. Oh, this is parallel. So yeah, parallel and convergent is tricky. If it's similar trait starting from similar ancestral, that's parallel. Gene environment interaction is when what responds to environmental variation in different ways. It's when two different genotypes respond differently to different genotypes. Okay, for now we're good. I'll see you tomorrow. So, peace.